Okay, so it's our pleasure to kick off this session now on patient involvement in sarcoma clinical trials. As you can see, I'm not Markus Wartenberg, but I guess you know me as well. My name is Bernd Kasper. I'm medical oncologist based in Mannheim University of Heidelberg, Germany. I'm one of the appointed board members in SPAGN, and I think I'm actually supporting this organization from almost the very beginning. I think I only missed the first meeting. And yeah, and my co-chair is Roger Wilson, so you can introduce yourself as well.
Well, I'm not sure that I need that much introduction these days, but welcome, and it's a delight to be here and a real pleasure for me to be working with Bernd. So let us proceed.
Of course. So the first, we will kick off this session with a talk, again, not from Robin Jones, which is actually here in the program, but it's my pleasure to introduce Emine Hatipoglu. She's actually a trainee at the UCL in London, but also affiliated with the Royal Marsden, so she's working together with all the well-known medical oncologists there, Sandra, Robin. So it's my pleasure to have you here, and she will give a talk on challenges of planning and conducting clinical trials in rare and ultra-rare cancers.
Hello everyone, it's a great pleasure to be here. I'm sorry Robin couldn't join us today, but I'll try to fill in the gap as much as I can. I'm a medical oncology trainee at the Royal Marsden Hospital, and I did my research in tumor immunology at UCL. So today I'll be talking to you about the challenges in clinical trials in rare cancers. I think first of all, the main question is how rare is rare, and what do we actually mean by a rare cancer or a rare disease? So there isn't a unified definition of what a rare cancer or what a rare disease means. That definition can vary from country to country. For example, in the US, a rare disease is defined as a disease that has a prevalence of less than 200,000 people. And it is estimated that there are over 7,000 different rare cancers, and they affect approximately 30 million people just in the US. So although it's rare, it's not actually a negligible number of people who are affected by these rare diseases. Across the world, it's approximately 10% of the global population is affected by some form of a rare condition. Despite that, there are only 5% of these rare conditions have an approved treatment option. So if you think about 10% of the global population, that actually equals to a significant number of people who suffer from the consequences of some sort of a rare condition. Because of the rarity of these diseases, we don't really know much. For a lot of them, we don't really know much about their etiology, and therefore we don't really have many effective treatment options. Therefore, recently there's been a great incentive to understand the etiology and the biology of these diseases to be able to develop better treatment options. So it's estimated that approximately 15 million people will have genomic testing of their cancer over the next 5 years to have a better understanding of their disease. From a patient experience point of view, I think it's important to understand how the patients feel in terms of the challenges they feel for like having a rare disease, having a rare cancer. So the UK Rare Diseases Framework has done a survey both with the patients and the healthcare professionals to understand what they feel are the main challenges that they experience. From a patient's point of view, what they found most challenging was that, first of all, awareness of the healthcare professionals about the rare disease was quite difficult and rare for them. So that was one of the biggest challenges. And because of this lack of understanding or lack of knowledge about the rare disease at the time of diagnosis, they felt that access to specialist care was a big challenge as a result of that. When they did the survey with the healthcare professionals, it was actually a quite similar picture. So the healthcare professionals felt that their awareness about rare diseases was quite low, actually, and that was a big challenge for them when they were faced with a patient who had a diagnosis of a rare disease. And that had a consequence on their ability to diagnose the disease and therefore refer the patient to the appropriate specialist center for treatment later on. I thought that was quite an important and actually shocking finding, because if we're going to talk about treatment options and especially clinical trials, the key thing is you have to get the diagnosis right, and you have to get the diagnosis right at the right point without any delay to be able to treat the patient at a timely manner without any delays. As a result of that, the UK Rare Disease group then came up with an action plan where they've said that we will aim to identify those patients with rare diseases as early as we can, and we will aim for a faster diagnosis to improve their clinical outcomes. They also have an agenda to improve the knowledge and awareness of the healthcare professionals about rare diseases so that the diagnosis is not delayed. And they are aiming to develop programs to accelerate the diagnosis and treatment of these rare diseases, including cancers. And of course, this whole journey has an important psychological impact in the patient journey, and they are aiming to take the psychological aspects of all of this on board and integrate it into the patient care. So rare cancers. So rare cancers is actually an umbrella term. As you can see here, there are many, many different types of cancers which are under the rare cancer umbrella. Even the tumors that you think are quite common, like lung cancer, can have subtypes which are classified under the rare cancer umbrella, and sarcomas make up about 5% of the rare cancers. So how do we develop new therapies for rare cancers, and how long does it take? This is a very generic diagram to describe the common pathway for developing a drug from the beginning to the end to the approval point. So first of all, to be able to develop any drug as part of a clinical trial, you have to identify a target in a disease. So you need to know what is causing the disease and what can you target in that disease to be able to control it. Once you do that, then you need to screen your different molecules, your different potential therapeutic agents, and that can take up to 5 years, sometimes maybe longer. And once you identify that specific potential therapeutic agent, then you need to go to the preclinical study stage, where you use animal models to kind of prove your concept that this actually is working in an animal model and can move to the human studies stage. Once you prove your concept in the animal studies, then you go to the clinical trials stage. So we start with phase 1 clinical trials. So in the phase 1 clinical trial, the ultimate aim is to prove that your new potential drug is safe. And you also try to establish the highest dose you can give at a safe level. Once you go through that stage and you've proven that your new drug is safe, you go on to the second phase 2 and phase 3 clinical trial stage, which is when you try to prove the efficacy of the drug. So you're trying to prove that your new drug is better than what is currently available or better than a placebo control. And if you have gone through all these hurdles, which takes approximately 15 to 20 years sometimes, maybe longer, then you finally reach a point where you can apply for approval of your drug to be standard of care. As you can appreciate, this is an extremely challenging and long process. And if you have a rare disease, which is potentially quite aggressive, you do not have this much time to sit around and wait. In terms of clinical trials that are available for rare diseases across the globe, this is, I think the specific numbers here is not that relevant, but this is just to show you the variability of number of trials available in different countries. So according to this graph, United States seems to be the leading country in terms of the number of trials they have available. It's followed by Canada and other European countries. But what makes me think about this graph is it also shows that when you have such difference in availability of clinical trials, it also means that there is a lot of variability in the patient's ability to access clinical trials, which is quite important when you have a rare disease and you don't really have any established or very many established standard of care options. So how are these clinical trials for rare diseases different than clinical trials for common diseases? I think one of the biggest and key differences is the patient number. Because the cancer is rare, you have fewer number of patients, which then makes the recruitment process for the clinical trial much more challenging, and it can mean that you need a longer recruitment process for the clinical trial, which can make it longer. And your site selections can be quite limited because you need to have a site where you have a specific patient population with maybe a subtype of a rare cancer with specific requirements about genomic subtype or treatment history. So that makes it a completely different process than running a clinical trial for a common type of lung cancer, for example. Also, geography can be different because you can't run clinical trials for rare diseases in any hospital that you want. You have to kind of follow where the patients are located geographically. The other important difference is that to run a clinical trial for a rare disease, you need to make sure that your clinical investigators and the research sites have the appropriate expertise to look after these patients and meet their needs both psychologically and in terms of all the medical treatment. And if they don't, they may need specialist training before the clinical trial, which can make it a longer process. So in terms of the top challenges for clinical trials in rare diseases, because of the number of patients, or the fewer number of patients that we have with rare cancers, you may need to open your clinical trial in multiple sites, which can mean that these multiple sites can mean multiple sites in different countries. And that comes with its own challenge because different countries may have different levels of experience in running clinical trials. And also the regulatory processes can vary from country to country, which can be quite challenging. Again, different countries and even different cities in the same country can have different ways of assessment, clinical assessment, different equipment, which can have an impact on how you run your trial and also have an impact on the clinical data you collect that can completely change your clinical outcome, your clinical trial outcome. So I think it's important to assess the infrastructure of the clinical trial sites that you want to open your study in and assess their expertise, quite important. Another very important challenge in running a clinical trial, especially in sarcoma, is the heterogeneity of the disease. So you can't just open a trial, say this is a sarcoma trial and include all comers. You need to have a specific trial for a specific question for a subtype of sarcoma, and that heterogeneity can be a big challenge in terms of setting up and running a trial. Oh, sorry, what did I do? I think another important factor in running a clinical trial in rare diseases is the pharmaceutical industry. So there is a concept of orphan drug development. So orphan drug is defined as a therapeutic agent that is used to treat a rare disease. And it is identified as a area, as an unmet need, because we do not have enough orphan drugs that we can use for drug development. And one of the main reasons for that is the lack of interest from pharmaceutical industry, which is mainly driven by financial incentives. I think this is a topic we can have long discussions about, because the ethics behind this is questionable. But regardless, this is the reality on the ground that we have to accept and find a way to deal with. So how do we overcome these challenges for clinical trials in rare diseases and rare cancers? So I think conducting feasibility studies before you open your trial is quite important, especially feasibility studies to understand how you can overcome the patient recruitment barriers is important, as it's one of the biggest challenges. Again, feasibility studies to assess the capability of this clinical site and the clinical investigator and their infrastructure is absolutely key, because you don't want to run into a trial, run into trouble while you're running into a trial, then you have to close the site later on. Another important factor in tackling these challenges, I think, is setting up effective collaborations between different sites, even between different countries, where you can pool your patient data, you can learn from each other's experience. And I think working with patient advocacy groups is absolutely critical in terms of making the whole experience as a more patient-centric experience, learning from the patient advocacy groups about what's important for the patients, and also that helps with patient recruitment as well. I think yesterday we talked about using new technologies to understand more about patient perspectives and patient experience, and I think that's quite important and applicable to clinical trials as well. Rather than having a very didactic way of running a clinical trial, it might be useful to make it a dynamic process where there is constant interaction with the patients and understanding what their needs are, what their expectation is, and what are the limiting factors for them in terms of joining a clinical trial, or what are the difficulties that they face when they join a clinical trial. What is really key really dear to my heart, and I think what is really important, is supporting research. If you do not understand the biology of a disease, it's impossible to develop an effective treatment or to consider running any clinical trial for it. Although, funding is always an issue in any kind of research, to be honest, but I think especially rare cancers such as sarcoma, research funding has been quite limited and is still quite limited. But I'm happy to say that over the past years, there's been more incentive to fund sarcoma research. It has been accepted as an area of unmet need by Cancer Research UK, so now they have dedicated sarcoma research funds available. Sarcoma UK has always been a big supporter of sarcoma research in the UK. But I think there is definitely a great need for more funding. The other key is how do you run your clinical trial? So the clinical trial design is very important in terms of having an effective clinical trial which actually gives you an answer to a specific question. Because of the patient numbers, running a randomized control trial like you run in a breast cancer may not be possible. So but you still have to go through that rigorous process that you have for a more common disease to be able to kind of have a standard of care in your clinical trial. So what do you do to overcome that? You need to have a very specific question, and you need to make sure that your methodology does not have any bias. So you need to minimize the bias you have. Also, you need to define your population that you want to include in the study and your subgroups. And you need to have very clear endpoints. So what are you objectively going to measure, and how are you going to do that? And also, when you have a small sample size, so a small number of patients involved in a study, you have to really do the maths right, if I may say that. So you have to use the correct statistical method that will give you an accurate result rather than skewing the result. There are different ways of running clinical trials that can be alternative, as we can say. Basket trials. So in a basket trial, you can have patients with different types of disease or different subtypes with a common unifying factor, for example, a common genetic mutation in a cancer, and you use one treatment for different types of cancer. Another one is an umbrella trial where you have a single disease, but you have multiple trial options in different arms. Or you can use a platform trial where you have multiple arms, and you can add or drop the arms based on your interval analysis. So these alternative clinical trial methods or designs can be used in rare cancers to achieve more meaningful results. The other thing to say about running clinical trials in rare diseases is that because it takes such a long time to run these clinical trials, there is an accelerated approval pathway that can be considered. I mean, there are different routes to do that, and I will not go through in detail because that's variable from country to country. But essentially, what we're trying to achieve with these accelerated paths to approval is that if you need to show that your data is sound and it is looking promising that the drug you have in trial is going to be more effective than what you have, and it is a safe drug. So the regulatory process for that varies from different country to country. In the EU, there is the PRIME system, which is basically, it allows you to start early communication with the regulatory bodies to speed up this process. And I think it's quite an important factor to consider when you have a rare disease. So has it always been bad? So do we not have any clinical trials? Do we not have any success? Of course we do. Over the past years, we have, I think some of my, yeah. So of course we do. So although we have had negative trials in different types of sarcoma, we've also had some success which has led to drug approvals in different sarcoma types. But I think there is still a lot of room for development. And more recently, with better understanding of the disease, and we've touched on this briefly yesterday, the TRAC fusions and targeting the TRAC fusions in different cancers, including sarcoma, with drugs like larotrectinib or entrectinib have proven to lead to successful outcomes in different sarcoma patients. So we did make progress, but the the challenges that I've just talked about still remain and they need to be tackled. So we need to address the rarity, we need to find ways of addressing the rarity of the disease and the heterogeneity of the disease. We need to find ways of addressing the variations in different clinical trial sites and the experience of the people running the clinical trials. We need to find ways of addressing the regulatory issues in different countries. And I think we need to kind of emphasize the importance of collaborations in different countries, in different sites. And I think it's important to take on board the experience of the patients and what matters to them. How can we make it easier for them to participate in the trials? Do they have any fears? What's the limiting factor for them? I think that's the end of my presentation. Any questions? Thank you.
Yeah, thank you very much, Emine. That was a very nice overview with already quite a couple of difficult trial designs and things. Yeah, I think we do have time for a couple of questions before we move to the second part of this session. If you want, you can join us here on the podium. So yes, please. Are there questions?
No questions.
Jane.
It's a topic that keeps cropping up about patient involvement in the design of clinical trials and how do you factor that in? Because, for example, my experience to date has been that you only get to hear about clinical trials when they're opening rather than having been involved at any stage prior to that. Any suggestions? I know it's a topic that's come up at a few conferences I've been to. I don't have an answer yet about where and when patients can be involved, or at least advocates on their behalf in the trial design.
I think it is a difficult question to answer. I think probably it's important to get that involvement before you set up the trial, so in the setting up stage, and I think the patient advocacy groups are quite critical in that. I don't know if you have any suggestions, but I think rather than getting them involved when it's already ready to go, that's because of also the trial regulations, once it's set, it's quite difficult to change. But I think the involvement needs to happen prior to and at the setting up stage. And I think maybe the pharma companies or the actual trial sites need to have it embedded as part of their trial setting up structure, that you need to have a representative from the patient advocacy groups, or you need to have, we talked about yesterday, apps maybe to kind of have that patient feedback beforehand to say, actually, I can't attend the trial because I live, 5 hours away, or I have a work commitment, and the trial visits needs to happen, every so often, or I have a fear of, going into a clinical trial because I think they're testing new stuff on me. So maybe having a clear communication, explaining what a clinical trial really means, understanding what the expectation is from the patient point of view, or what their fears might be to start with, might be a way of overcoming it, but I don't think there's a clear-cut answer.
I wanted to give you an example of failure where the patient could have changed this if we organize this correctly at the beginning. The example is a trial we did with URTC about metastatic patient, metastatic GIST patient who receive Glivec, and we wanted to randomize the surgery to know whether surgery in such situation improves the survival or not. And in fact, we had to stop this URTC trial after 2 years because we include in 2 years, maybe 10 patients, something like that, because the patient didn't want to participate, because they say, I am sent by my doctor who said I should be operated, I want to be operated. And you had other patients who told me, if you don't know if surgery is efficient or not, I don't want to be operated. And then we discussed with patient association of GIST, and the patient association said, of course, it's not well accepted. This trial, the design is completely unacceptable. And for me, it was really a lesson, because I understood that when you do a trial, the first thing, the design, you must discuss acceptability by the patient. And for me, of course, it was a failure, but I learned something. And I learned that we should have discuss acceptability of the trial with the patient to do another kind of design. We could have asked the same question, but with another design that just to randomize the surgery, yes, no surgery, no surgery.
Sylvia's picked up an example which we were going to use later in our discussion as well. I remember that study very clearly, and I remember being very angry when I saw the protocol, because I knew that no patients had been consulted. So it was a very, very important lesson for the sarcoma community. And I have to say that that study is the reason that EORTC, and partly guided by Bernd when he was chair of the soft tissue and bone sarcoma group, the importance of having patient involvement in the development of EORTC clinical studies. Now, if every other development organization, every other academic unit, every other pharmaceutical company followed suit with that, we'd be in a very different position. We'd be much closer to where I know you want to be, Jane.
This study was clearly a failure, but all the groups, including EORTC, clearly understood that the patient must be involved from the beginning of the design of the study. So it was a failure, but it was a lesson.
Now, I agree with you completely, Sylvie. And you know, one of the great pleasures for me was was part of the working within EORTC to help make that happen. EORTC now has a patient panel and has patients in every one of its study groups, and they come from all over Europe. Of course, the main requirement is that they can speak English, because that is the language of the science. But it's been a huge step forward, that study. It was very important, as Sylvie describes.
May I ask one last question? You've underscored the rarity the rarity or the challenge doing trials in rare diseases compared to common diseases. And I'm wondering if, when we look at the design and the acceptability by regulatory authorities, is there an opportunity to kind of engage with them to look at, because you showed that very long timeline, and you know how long to get from discovering something to getting it approved. Are there some opportunities for overcoming challenges by discussing designs with the regulatory authorities and what they might accept, especially given the uncommonness of the disease?
Yeah, I think it's something that's important. They have different pathways. So once this study has happened and you have preliminary data, you can try to accelerate the approval process. But I think because it's there is such a great need for new therapies and more number of clinical trials, I think all steps of the clinical trial pathway can be revised or should be revised with the appropriate organizations and regulatory bodies to kind of make it more effective for the patients. But I'm not aware of any flexibilities as such at this stage in time yet.
Okay, so one, Markus, okay.
I would like to make a short, more general comment. I think one issue is patient involvement in clinical trial. We are doing a lot in GIST and sarcomas on a national level, but also on an international level. But this is a general approach. This is not only limited to GIST or sarcomas. This is general, a topic in cancer or a general topic in medicine. And this is not new. 40 years ago, we have seen patient involvement in research in the HIV/AIDS community. So we are talking since 40 years about involvement. And in cancer, we are doing this now for around about 20, 25 years on an international level in different countries. And for our experience, the UK is quite far ahead in public involvement and patient involvement in research. But it is on us as patient advocates to drive the agenda also on a national level, together with other cancer organizations, to be involved in research as early as possible. And this will need a cultural change within the next years, that also doctors, but also pharma companies understand that this is crucial to involve the patients as early as possible into the whole process. And let's look to other industries. When you look to the automotive industry or maybe the software industry, it is absolutely mandatory to involve the customer as early as possible into the research process. And many of these industries, they talk this co-creation. They invite the customers to co-create the design or the development process. And we need to overcome this, and we are doing this in a very different level from country to country. We just started with this process in Germany 3-4 years ago, and we're making a lot of progress now with the government and with different other cancer organizations. But for my feeling in the moment, specifically also when we talk about personalized oncology, when we talk about precision oncology, but also about being more patient-centric, then we need to drive this forward from a patient perspective, and I'm sure we will also come to this more closer and more often within Sarcoma Patients Euronet, but also within the Sarcoma network, but also in training and educate our colleagues within the community. But the general approach is we need to fight for this, we need to drive it forward on an international level, but also on a national level.
Okay, last comment or question, then we have to move on.
I have a comment similar to what Marcus was just saying, and also what Jane had said previously, going to several meetings and continuing to have this conversation. I know many of us in this room are tired of having the conversation of, we know we need patient involvement sooner. We absolutely know that. And we continue to have this conversation. And I agree with Marcus, this is something that we have to drive the change. But, you know, we have pharma partners in the room. And part of the issue as well, and our medical professionals who all say we need to have patients involved sooner. We know that outcomes are better when we have patients involved. We know clinical trials have better recruitment when we have patients involved sooner. But we continue to say it, but we don't do it. And yes, there is some level of we can force or we can advocate for our governments to include patients. But we also really need our medical professionals and our pharma partners to push for that within their own areas as well. Because we are not going to be able to do it unless they take on that charge and say, we're going to push for it too. And it has to be something that we stop talking about and we actually start doing.
Well said.
I agree. Thank you.
Okay, so thank you very much again. So we move to the second part now, and it's Roger's task. And it will be much more interactive. I promise.
And if you want to interpret what Bernd said as a threat, you're entitled to. You've just had three very, very good presentations with a lot of detail, and you're sitting, and I'm sure you're aching to stand up. So this is where we open the secret. You've all got your envelope. Yes? All got your envelope? If you haven't got an envelope, Sheila's got one at the back if you need one. One needed there, Sheila. Thank you. Right, you can open your envelope, and in it you will find a coloured piece of card. Don't worry about the piece of paper for the moment. Worry about the piece of card. Find the piece of card. Put the piece of card in your right hand. Hold it up. Red, blue. Ooh, a lot more blues than reds. Oh no, there are plenty of reds. Good, good. Now, stand up. Put your hands above your head and clap twice. Now, those of you with a red card, sit down. Blue card, stay standing. Right, blue card, do it again. And now, stretch your hand up, reach for the ceiling. Reach, stretch, come on, stretch. Put your hands down and sit down. Now, the piece of paper is a form, which should only take you a few seconds to fill in, but please fill the form in. And then Mickey and Catherine will collect them. Oh, sorry, I didn't realise. Thanks, Bernd. Right, on that form you'll see we're asking you to just tell us whether your gender, give us your year of birth, and then 4 questions. Did you stretch up and reach the highest you could? Now, if you're disabled and were unable to reach up, do say no, because that just gives us a little bit more information. Did you find this embarrassing? Scale of 1 to 4. Do you have any after effects from doing this exercise? And how good do you feel having done it? Okay, right, let's have your forms in, please. Can you put the slide up? The slide, it's a single slide with a coloured background. Yeah. Just put it up. Lovely, thank you. Okay, all forms in. Just a few more over there. Right. Now, one person, when she came in and I gave her an envelope, actually guessed a bit of what was going to go on. Mark you, she's a scientist and a researcher, so I'm not surprised. But she said, I'm being randomized, aren't I? So I said, yes, you are. So how many people realize that you were randomized in that little experiment? Ooh, a couple, good, that's nice. But professionals, I notice. Now, we've got some data from you. You notice we didn't ask for your name, so we're not doing personal identifications here, but we're going to do some analysis, simple analysis to start with, of the answers you've given. And of course, what you really want to know now is what was our research question? What are we actually trying to find out from what you've done and what you've told us? Well, our research question is the embarrassment of reaching up with your arms in public mitigated by the beneficial feeling you get from doing it? And we've got two groups, one of which only put their arms up once, and one of which put them up 3 times. It'll be very interesting to see if we do get a difference. If we don't, we don't, but if we do, we do. That's the chance we're taking with something really which is quite unstructured. But as a patient looking at a clinical trial, and ideally looking at it, as Jane was saying, from the very early days, from the very early stages, and you look at the design, does the design answer the question? So let's just think about how you might ask questions if you can get involved with the design of a clinical study. We start with the research question. Is it the right question? Is it a question which patients see as valid? Now, the example that Sylvie gave of the GIST surgery trial, patients did not see it as appropriate for them. So that appropriateness and that validity are two very, very close, have a very close relationship, very close together. Now, who should the question be asked to? And in what way do you ask it? We've had mention from Emina of basket trials and umbrella trials. So we need to understand basic methodologies. We don't need to understand the detail, but we need to understand the principles which lie behind those methodologies and how they should be used. And we need to consider what might be appropriate experimental arms and appropriate control arms in a randomized study where you're making comparisons. And as you probably know, in science, a randomized study, especially if it's blinded so that nobody knows who's been selected into which arm of the study, has the greatest scientific validity. So, that's the start. The research question and the underlying design. But then we move on to those important issues that come through in a research protocol. When I've been asked to look at studies early on, one of the first things I do is to consider the inclusion and exclusion criteria. I can remember in the very early days of cancer research and patient involvement in cancer research in the UK, more than 20 years ago, one of the exclusion criteria we saw very commonly in clinical trials of new drugs in advanced cancer was to exclude people who were aged over 65 or over 70. Now, cancer patients tend to be older people. So why exclude people over 65 or over 70? And we went into our research meetings and we asked those questions directly to the doctors. And it came out fairly quickly that it was habit. That's all it was. They'd always done it that way. So that limitation got dropped because of patients asking questions about it. And if you need to exclude people on the basis of frailty, today you do it by selecting on a basis of performance status, which, there are very good tools for assessing the performance status of the functional status, if you like, of a patient and whether they're likely to be able to respond properly in a clinical study. And that's just one example of how patients can begin to make a difference. It's a very simple one, and it wouldn't apply today because that exclusion criterion has gone. But those are the sort of things we can do as patients. Now, the next area I always look at are endpoints and the outcome measures. Now, an endpoint will be how the trial is going to end for the individual patient. Where does it stop? Now, the trial might have treatment, if it's chemotherapy, good old-fashioned chemotherapy, such as the clinical trial I had. There were 6 cycles. That's when it stopped. But the end point was actually nominally 5 years later. Was I going to be a survivor after 5 years? And in order to track me during those 5 years, the theory was I was going to have a CT scan every 3 months. Now, no one today would ask a patient to have a CT scan every 3 months for 5 years. You would not put that in a protocol because the amount of radiation you're being exposed to is enormous. A single CT scan has the equivalent radiation of about 400 X-rays, standard X-rays. Now, the idea of having, however many it is, I can't do, 4 by 5 is 20. The idea of having 8,000 X-rays over 5 years is crazy. But that was in that protocol, the one that I was on 20 or so years ago. No one was there to question it when it was in the design. And that was an EORTC study I also hasten to add. Now, the outcome measures are the way in which the trial is reported. Now, the standard outcome measure, which is regarded as the gold standard, is overall survival at a time point. So it might be 2 months after completing treatment, or it might be 2 years, or it might be 10 years. Obviously, if it's 10 years, the trial is going to remain active for 10 years, and there's going to be a cost in that. So that sort of overall survival endpoint is rare. But we also have confounding factors. If you have a study of something like chemotherapy, and patients relapse after 6 months or a year, they will then go on to another therapy. They'll go on to a second line of chemotherapy, perhaps, and then maybe a third line. And that makes, obviously, in the study with overall survival, a primary endpoint, it confounds it, because some patients will have had first line only, some will have gone on to second line, some will have gone on to third line, and they won't be directly comparable. So we have what are known as surrogate endpoints, which are closer, which are closer to the time frame to what the patient is experiencing. So you might have progression-free survival at 3 months as an endpoint for a study. And there are many, many different surrogate endpoints. A surrogate endpoint needs to be a validated one that's been used before and shown to be statistically and clinically to be appropriate. So there is another thing we can ask about. We can challenge. We can say, is this surrogate endpoint the right one? Should we be looking at response rate rather than survival as a key measure? You may, I would add, agree with everything the researchers have put forward, everything they've looked at, everything they've considered. There's nothing wrong with that. But do ask questions. Now, let's look at the treatment itself. A treatment schedule in a clinical trial requires visits. Nobody enjoys visiting hospital. Nobody enjoys the experience of finding a car parking space. Nobody enjoys the railway journey. How many visits do you have to make? How frequently? When you have a visit, you might have tests. I can remember looking at a draft trial protocol, which was in breast cancer, and it was suggesting that the patient would have a biopsy every 3 months for a year. Now, biopsy is intrusive and it's painful, even with good anesthesia. No one wants to go into a trial with 4 biopsies in a year. So, we could question that, and we could say that was inappropriate. And that was a young oncology doctor in training, and he came up with that idea during a session on designing clinical trials. And it was very useful because it got the whole cohort talking about the issue of tests. But we naturally think about tests. We've undergone them. I don't mind a blood test, but I don't want 3 in a day, for example. Now, side effects. This is a big one, of course, because the earlier the stage of a trial, the more likely there are to be unknown side effects, ones that the doctors haven't been able to plan for. So if you're looking at a very early phase trial, side effects is a big issue to consider. Now, the management of side effects and how they're actually controlled or mitigated or prevented is an area where we have a lot of input in a clinical trial. Getting the knowledge of what side effects are expected, what are possible, because a new drug belongs to a class of drugs, and generally speaking, in a class of drugs, there are similar side effects between different agents. And such issues as dose reduction, or even stopping treatment, are ways of mitigating side effects. Clearly, they have an impact on the way the study performs, but the issue for us is how it works for the patient. We don't want a patient harmed. I'll come to a key question on that issue in a moment. Then there's follow-up. After treatment, what sort of follow-up is proposed? Now, Sylvie and Jendrik, as surgeons, talked about follow-up within their talks, but there are follow-up issues for every kind of treatment that we might have to experience. And what tests will there be during follow-up? That's relevant as well. Now, if treatment fails, acceptability of tests, there we are. Now, if treatment fails, what happens next for the patient? And that's relevant too if treatment succeeds. If you're cured at the end of a clinical study, what happens next? You walk off into limbo. Being cured is an odd experience. I've been in remission for 7 years once, and I'm currently in remission for coming up 11 years. And some people might think of me as cured, but I feel more comfortable not being cured. I feel more comfortable being seen by my oncologist once a year and having an X-ray, a check-up on me, and a chat with him. Makes me feel much happier. Those sort of things need to be cared for and thought about in the way a clinical trial is designed and managed. And then there's 2 questions which I regard as 2 questions of balance. And the first one is benefit versus burden. Step back. For the patient who enters a study, we need to think about benefit versus burden. That came through very clearly in the surgical talks. Sorry, done that again. In the surgical talks. These experienced surgeons know about this issue of benefit versus burden, and they know about it as a long-term issue. I've been very grateful. I've had an amputation, as most of you know, and I'm very grateful to a surgeon who knew what the issues were, who ensured that I could talk to other people who had lost a limb before I made the final choice of going down that road and having the amputation. So good surgeons, regardless of a clinical trial, are looking at that benefit versus burden equation. And we need to learn about it. We come at it from a different angle, and we should be coming at it from a different angle, but we need to learn about it and use it. And then the final question, which always needs answering. Would you enter this study? Now, if you were in the position of this patient, you may never be likely to be in the position of the patient in the study that you're looking at, but would you enter this study? Now, it's 11, I don't never got you. No, it's 10 items there, aren't there? Isn't that neat? No, there are 11, beg your pardon. There are 11 items there, I never counted them before. But they're a very nice, simple, short guide to the kind of questions you can approach looking at a clinical trial from. There are others, particularly where you have lived experience yourself, and a trial is relevant to your particular lived experience. Now, let's address the question that Jane raised and that Marcus touched on as well. We need, as an advocacy community, to empower us. To empower ourselves to be involved. And if we can equip ourselves as an advocacy community to be available to pharma, to academic doctors looking at research, to our universities, if we can make ourselves available, the pressure that is growing from regulatory bodies and ethically for patients to be involved in a study is really, really strong. And we can make it happen. It's not going to be quick. It's not necessarily going to be easy. But if we are able to do that, and I've been talking with Catherine about how SPAGN might be able to build a register of patients, those with lived experience, and experienced advocates who understand the issues that an individual study may bring up. If we can do that, I think we can move this agenda forward. But I've been doing involvement for 20 years, more than 20 years, such that the very first research meeting of professionals that I went to in the UK back in 2002, every single doctor in that room at that time has since retired. And I'm still around and still asking questions. So, to me, it's a great delight to see people like Amina coming through, into the community, clearly very committed and very able and taking the banner forward. And I want us to be there, empowering her and empowering others, empowering Sylvie, empowering Uta, empowering Jendrik, as people who are important to us, and we can make it better together. Collaborate, collaborate, collaborate. So, thank you. Questions?
Thank you very much.
You've got a result.
Yeah, we have the results.
Put the slide up. We have the initial result. Right. Those in the blue group, 41 in each. That's very nice. Did you find this embarrassing? Score of 1.27, score of 1.23. That's interesting. Just marginally. Mark, you 0.4 when, 0.04 on a total score of 4 is what? 1%. Yeah, okay. I was hoping that the blue might be a little bit higher, but there we are. And how good did you feel? Is it? No, it's an average. It's an average, not a percentage. How good did you feel from having this extra bit of exercise? Well, obviously those on the blue group felt a lot better than those in the red group. So those extra 2 arm stretches made a difference. Brilliant. Thank you very much indeed.
Thank you very much. Are there any final questions?
How you doing? It seems to be that a main block for all of this is patient involvement. But as a patient, what I first did when I was diagnosed was I went to social groups, like social medias and stuff like that, to kind of gauge a bit more information. Has any consideration been given to that, to utilise that sort of network of data?
Yes, I'm sorry, I didn't quite pick that up. Difficulty of moving while listening. 2 things at once, it's getting beyond me.
Yeah, social media seems to be a main outlet for myself to get a lot of information regarding the disease, and patient involvement in studies seems to be a bit of a roadblock. Has any consideration been given to utilising that data that's available on a global network?
Oh. I'm not aware of it. I'm not a great fan of social media itself, I must admit, but that's a generational thing. But I do know that social media can have an influence on the way patients think and feel about a clinical study, but I'm not aware that it's helped create better studies, because that ultimately is our objective. It's better clinical studies. Studies that patients will enter, studies that will accrue quickly, and studies which will deliver results quickly. Now, social media has a little part to play, certainly, in, I think, keeping people engaged in a study, particularly a long-term one, but I'm not sure that at this moment I can see a role for it beyond that.
Sylvie.
Yes, for all these medical trials, in fact, the objective is to have a better survival, better results. So finally, the acceptability, except for the example I gave, is rather good. But there is another field of research where the acceptability is, in my experience, difficult. It is genetic counseling. When you suspect that a molecular anomaly, constitutional molecular anomaly, is pre-existing, and that you propose to the patient genetic counseling to test if there is a genetic anomaly which can give another cancer, for existence. I have had a lot of patients who refused because they don't want to know whether they have the risk to develop another cancer. So it is, in my experience, it is the field where it is the most difficult to have the acceptability of the patient. I don't know, Bern, if you have another experience about that.
No, he's not. I think you're right, Sylvia. The underlying issue here is that research is not really understood by the general public. And I think the purpose of research is actually something that we need to do more in the education system, and we need, as an advocacy community, to do more to promote with the people we talk to and talk with. You know, the value in research is obviously something which, for me, being involved in it, I'm doing altruistically largely. I'm hoping to get benefit from it, but I may not. But I'm hoping that the bigger picture to which I'm contributing actually does deliver benefit for the future group of patients. And that part of the message about research is quite powerful for people. But it's not something that a doctor should be promoting because it's, you know, it's outside your, if you like, your ethical remit to do that. So, but it's something we can do as an advocacy community to help accrue patients into studies.
Okay, I think we close the session then. We will have a coffee break now, and I just want to draw your attention that after the coffee break we will split up into 2 working groups. There will be one working group on clinical trials, which is actually here in the room, in the auditorium, and the second working group on sarcoma awareness is just opposite in room 4C. Enjoy your break. Thank you very much.
Cool.